Hi guys, today I want to jump right into the part 3 of this enzyme video series, the Michaelis-Menten kinetics and the Lineweaver-Burk plot. So this is really not just for MCAT. If you're here for your biochemistry class, you're at the right place. Okay, so there are 4 inhibitions that I want to talk about today, as you can see. And then first we're gonna talk about this Michaelis-Menten kinetics again. This equation, okay? So this VO equals Vmax times S substrate over Km plus S substrate. In terms of MCAT, I don't think you need to necessarily memorize this equation. I've never seen MCAT ask you to plug in the numbers and do some math. But what we derive from this equation is very important, that we know Km is inversely related to the enzyme and substrate affinity, right? If you have a high Km, which means the enzyme and substrate affinity is pretty low, right? Whereas if you have a the enzyme substrate have a high affinity, then your Km will be relatively low, right? Okay, and then that makes sense because once again, Km is the concentration of the substrate at half Vmax, right? So if your affinity is really strong, then you really don't need that many of the substrate to saturate the enzyme, right? Because they just don't come off. They just stick on it, and then the reaction happens, right? So it's just easier to saturate the enzyme, so the Km will be relatively low if the affinity is high. All right, so now let's move on to the 4 inhibitions and then their corresponding graph. Okay, so competitive inhibition is the first case we want to talk about. So competitive inhibition is when you have some inhibitors that we will indicate with the letter the inhibitor I that will bind to the substrate's active site that competing with the substrate, because the active site belongs to the substrates, right? But now some inhibitor that have a very similar structure with the substrate binds to the enzyme and form the EI complex, which stops the formation, not stops, but inhibits the formation of the ESI complex, right? All right. There are 4 things we need to know regarding each inhibition, okay? So the first thing you want to know is, where does this inhibition happen? Where does competitive inhibition happen? So it happens at the active site of the enzyme, right? All right, so the second thing we want to know is how the Km is affected. And then the third thing we want to know how the Vmax is affected. And then lastly, we want to know how do we overcome this inhibition if there's a way to overcome it, okay, to begin with. All right, so how does the Km change when we have competitive inhibition? We can actually look at the affinity, right? The affinity of the how does the competitive inhibition change the affinity, and then we can just tell, how does how does the Km change accordingly? All right, so as you form more and more EI complex, that stops the formation of ES complex, right? So I said affinity, what is the affinity? The affinity is the affinity between the enzyme and the substrate, the enzyme-substrate stickiness, right? So if the affinity lowers in this case, so the Km would increase, right? Okay, so that's how Km is affected, okay, when there's competitive inhibition. And then what about the Vmax? So first, let's think about if this inhibition can be overcome by any means, right? So what about I increase the concentration of my substrate, right? That can actually outweigh the effect of the inhibitor, right? That can be actually kicked out, and not kicked out, it's still in the system, but it won't win the race of binding to the enzyme. So eventually this can be overcome by adding more substrate, right? So if the if this can be overcome, then my Vmax really doesn't get affected. My Vmax will stay the same, right? As long as I add enough of the substrate to stop the competitive inhibition. Okay, so to reflect it on the graph, I'm gonna draw a graph without any type of inhibition. Okay, this is the original status, and then once I have this competitive inhibitor involved, as you can see that my Vmax will stay the same. My Vmax will stay the same. But what about my Km? Again, Km increased, right? So to show it on the graph, it would be something like that. Okay, so this Km, this new Km will be here. Okay, what about the original Km? The original Km will be here, right? They have the same half Vmax. They have the same half Vmax, and then this is the original Km. See, my red Km after the competitive inhibition increased, right? It shifted to the right. Okay, and then my Vmax is same here. Okay, all right. So now let's move on to uncompetitive inhibition. Okay, so uncompetitive inhibition, unlike the competitive inhibition, it doesn't inhibit the active site of the enzyme. It actually waits until the ES complex is formed, and then this inhibitor will bind to the ES complex and then form the ESI giant complex. Okay, so the location of this inhibition is at the allosteric site. Allosteric site. Okay, what does it mean? Let me draw you a picture to show you visually. Okay, so let's say this is my enzyme. Okay, so I form this ES complex, right? And then allosteric site is the site where it's not the active site. Okay, so let's say my sorry about that. My allosteric site is here, right? So the inhibitor can bind to, let's say, here, right? So once the inhibitor binds to the allosteric site, what does it do? Like, it can kind of lock the enzyme to prevent the release of the product or substrate. If you, yeah, I mean, I'm not sure if with the inhibitor binds to the allosteric site, if the product will even be formed or not. But even if it can be formed, the enzyme kind of locks the product. It doesn't get released. So it eventually prevents this step, right? Okay, so the second thing we want to know is how does it affect Km, and then how does it affect Vmax, right? And then if it can be overcome by any means, right? Okay, so thinking about Km, let's think about affinity again, okay? So what do you think about affinity? This is actually pretty tricky, right? You might think, oh, ESI, more and more ESI are formed, so which means I have less ES. Well, not according to Michaelis principle, right? According to Michaelis principle, I mean, I never got rid of the substrates, right? So the reaction ES is still binding to the enzyme. Substrates are still binding to an enzyme. So the system would think, oh, where's my product? My product ES complex. I don't see that, right? So I'm gonna push forward the reaction toward the ESI side, according to Michaelis principle. So this is actually increasing the affinity between it actually increased the affinity of the ES complex, right? So which means the inverse relationship of Km and affinity tells us, in this case, the Km will go down, right? Okay, what about the Vmax? The Vmax will also go down, right? Because this step gets prevented. So, I mean, this ESI, really there's no way you can just saturate the enzyme as without the inhibition, uncompetitive inhibition, right? Okay, so let's draw the graph. So once again, this is the graph before the uncompetitive inhibition. And then with the uncompetitive inhibition, we know that the Vmax decreased. So Vmax will be lowered, right? And then the Km will be decreased as well, right? So to reflect it on the graph, my Km, new Km will be here, right? But the original Km was, let's say, that's a little bit far stretching. Let's say it's here, right? So you can see the decrease of Km on this graph. All right, so that's the case of uncompetitive inhibition, okay? Now let's move on to mixed inhibition, okay? What is mixed inhibition? So as you might have guessed, a mixed inhibition is a mix of competitive inhibition and uncompetitive inhibition, right? So we have inhibitors binding to the active side, not active side, the enzyme, but not at the active side in this case, right? It will be both binding to the allosteric side of the enzyme, okay? All right, so when we know that when this case happens, when EIs are formed, we know the Km would go up, right? So the Km would go up, of course, when the EI is formed. And then also we see that the uncompetitive inhibition, the Km, oh, sorry about that. The Km goes down, right? So to think about, so what happens to the mixed inhibition is if you have a more EI formed than ESI, then your Km will follow the path of competitive inhibition. It would go up, right? If the ESI gets formed more than the EI, and then the Km would go down, right? Okay, just to color code this. And then this is the case like competitive inhibition, okay? And then what about the Vmax? Right? The Vmax would go down because these steps happen, right? As long as this inhibition happens, you prevent the formation of the product, then the enzyme Vmax decreases, okay? And then there's also no way to prevent this, okay? And then this situation, we don't draw the graph since it's dynamic, okay? All right, so now let's move on to the last case, the non-competitive inhibition, okay? So as you can imagine, non-competitive inhibition is actually is a special case of mixed inhibition when you form 50% of EI and then 50% of ESI. Okay, the quantity equals to each other. And then in this case, the Km that got brought up by the case of by the yellow case is balanced out by the decrease of Km that caused by the blue case. Okay, all right. So, what does that tell us? That tells us the Km actually stays constant in this case, right? What about Vmax? Once again, the Vmax would decrease, right? Since this steps happens, right? And then they both bind to the allosteric site, and then there's no way to overcome that, okay? And then this we can reflect it on the graph, okay? And then this is the graph without any inhibition. And then when non-competitive inhibition happens, you know that Vmax decreases, right? And then what about the Km? The Km stays the same, right? So it'll be something like that, okay? You have a lower Vmax, but your Km okay, see, they have a constant Km, right? But a lowered Vmax. All right, so that's the 4 cases of inhibition, okay? You just really want to know how does each inhibition affects the Km and Vmax, okay? And now let's move on to the Lineweaver-Burk plot. All right, so I'm gonna clean up my study guide a little bit. Okay, so as intimidating as this equation might seem like, you can actually tell this is just a flip of this equation. right? And then after you flip it, it's really just y equals mx plus b, let's say, right? Why? Because you can see that this y here is really just 1 over v, right? 1 over v, the y-axis. And then the x-axis is really just 1 over S concentration, right? That is the x. Okay, so it's really not that bad. And if you do the math, you calculate the y-intercept and then the x-intercept, you know that when x equals 0, y equals to 1 over v max, okay? And then when y equals 0, x equals negative 1 over km, okay? So let's draw a graph without any inhibition, okay? So that's my graph without any inhibition, okay? So now let's talk about the case of a competitive inhibition, okay? Let's see how the competitive inhibition reflects on the Lineweaver-Burk plot, okay? So as you know, my Vmax stays the same, right? Here, my Vmax stay the same, right? So Vmax stay the same, which means what? Which means 1 over Vmax will stay the same as well, right? All right, and then what about my Km? So my Km increases when there's competitive inhibition, as we discussed. So what about negative 1 over Km? It's a double flip, right? First you have a negative sign, and then you have a 1 over. So that's really doesn't change. So which means Km increases, negative 1 over Km would increase as well, right? That would bring negative 1 over Km to the right because it increases, right? Okay, so if I were to plot this, that's the graph after competitive inhibition, okay? Okay, easy. So now let's draw the case for uncompetitive inhibition, okay? So once again, the y-intercept is 1 over Vmax, and I suggest you to memorize 1 over Vmax as the y-intercept, and then negative 1 over Km as the x-intercept, so you don't do the algebra on your test, okay? During your test. Okay, so to draw a line without any inhibition, this is what this looks like, right? Okay, now once again, how does my Km change? So my Km would decrease in this case, right? So my Km decrease. What about negative 1 over Km? It would decrease as well, right? Double flip. So if I decrease negative 1 over Km, then it shifts to the left of the axis, right? Okay, what about Vmax? So Vmax in this case decreases as well when there's uncompetitive inhibitor. So 1 over Vmax would what? Would increase, right? All right. So let's say okay. That's the original 1 over Vmax. So after the increasement, then 1 over Vmax will be here, okay? And then if I were to draw the plot, it looks like that, okay? It is parallel to the original line, okay? All right. So let's do the last one, the non-competitive inhibition, okay? So once again, not to sound like a broken record, 1 over Vmax. And then let's say this is negative 1 over Km, okay? So I plot a line like that, okay? Okay, so what about Km? Okay, Km in this case doesn't change, right? Km is constant. So negative 1 over Km will be constant as well, right? So I'll be here. Okay, what about Vmax? So Vmax in this case, it decreased. So 1 over Vmax would increase. So it will be here, right? Okay, so this is the line after non-competitive inhibition. Okay, so I just really want to drive home the point that it might make all make sense when you look at me drawing, watch me drawing all the different scenarios, but I want you to just do this practice, like when you see, okay, the white line is the non-inhibition case, and then the A, B, C, there are different types of inhibition, and then can you tell right away what is A, what is B, what is C, right? And then also with the Lineweaver-Burk plot, A, B, C, what is the case for A, for B, for C, okay? Just train your brain to instantly see this, and then you know this, okay? All right, and then another thing I really want to touch base on is the sigmoid graph. I talked about this briefly in the part 2 enzyme video, but I actually misspoke something, so I want to just correct that here, okay? So the sigmoidal graph that you see, as I discussed in the last video, is when there are multiple binding sites for the enzyme, right? So the substrate can, because there are multiple binding sites, the substrate can either promote or inhibit the binding of another substrate to another binding site, right? So this is called cooperativity, as I discussed in the part 2 video. So, and then we also said like the Hill's coefficient reflects if it's a positive cooperativity or negative cooperativity, right? If the Hill's coefficient here, Hill's coefficient is larger than 1, then we know it's a positive cooperativity, right? If it's less than 1, then it's negative. And then if it equals to 1, so which means there's no cooperativity, which means there's no multiple binding sites, okay? So the mistake I made is I showed you the graph that without any cooperation, yes, this is Hill's coefficient equals 1. That we've drawn this many times, right above. This is without any multiple binding sites. But when it's positive cooperative, then the sigmoidal line is like that, right? But the negative 1, I drew it like that in my last video. I just want to correct that. It's supposed to be like that, okay? This is the case of negative, you have the negative cooperativity, okay? Yeah, this actually kind of looks like the case of uncompetitive inhibition, okay? So sorry about the misspeaking. I hope that's helpful.